All right, next video being recorded right after the first. So, surprise. Let's jump right in. This is going to be regulation of gene expression. So, how do we decide what genes to make and when? This video is going to focus specifically on prokaryotic gene expression. Next one will be eukaryotic gene expression. And then we have mutations and lab techniques. It's going to be a lot of lectures this unit. Looking like we have this one and this one and three more. So let's get into it. We've got regulation of gene expression. So what do I mean by gene expression? I mean the process where a gene is actually used to make a gene product. Just having a DNA sequence doesn't mean we actually make the protein that the gene codes for. So it may not have any actual effect. So gene expression is kind of like, to what degree is this gene being used and what effect does it have? Most often this product is a protein. We've kind of gone along so far just assuming it's a protein, but it could also, RNA is sometimes the final product. It can be just regular old RNA. It could be tRNAs have to be made. There's genes for tRNAs, there's genes for ribosomal RNAs, and there's other types of functional RNA that exist. So the product of a gene is not necessarily a protein. It's often a protein, but not always. And we'll talk about those other types of functional RNA a bit in, I think it's the next lecture. So then gene regulation is how our cells control that expression of those genes. So how do our cells decide when to turn the gene into a final product versus just leaving it be? There's a couple of terms I'm going to use a lot. First is regulatory sequences. If I say a sequence especially, that means a DNA sequence or RNA sequence, depending on what it is. And these are non-coding, so they don't code for a gene, but they're DNA sequences that regulatory proteins will bind to. Think a promoter sequence or the TATA box as an example, but it's essentially just attachment sites. And then regulatory genes are DNA sequences that actually code for regulatory proteins, like we mentioned in the sequence definition, or other regulatory RNAs. So these actually code for proteins or functional RNAs, but they their only job is to regulate other genes. They don't have a function outside of that. One of these RNAs is called small regulatory RNA. We can't use rRNA because that's already reserved for ribosomal, so we go with sRNA for small. And these are just RNAs that will bind to the mRNA strand using complementary base pairing, and they will either activate or inhibit translation. So a lot of this next section just really has to deal with, like, how does the machinery of transcription and translation attach to the DNA or RNA strands to actually do their jobs? And how can we make it easier for them to attach, or how can we make it harder for them to attach? So these are the main components kind of laid out. You have your gene in a DNA sequence, and then you have a regulatory sequence, which is right before it. This is like your promoter sequence or inhibitor sequence. This is where regulatory proteins will attach to, or maybe even to, the RNAs itself. But the sequence is right next to the gene, whereas the regulatory genes themselves, the ones that encode proteins, they can be anywhere. They can be on the same chromosome. They could be on a different chromosome. They're not necessarily right next to the genes they regulate. And again, they can either produce a protein that'll bind to the sequence and do something, or a small RNA that will also bind to the mRNA sequence and stop something from happening or promote it happening. So basically everything we're going to talk about, they'll have slightly different namings, but they will all fall into one of these categories. So let's talk about regulatory proteins. Regulatory proteins are ones that control the gene activity. They basically increase or decrease transcription. what we have called these transcription factors in the past as well. You'll see that as another name for them. They come in two flavors. The first is repressor proteins. These are proteins that when they bind to the DNA, they block transcription. So they often just make it impossible for RNA polymerase to come in and attach. Activator proteins are their other half. They stimulate transcription when they bind to the binding site, that regulatory sequence. So an activator protein might be like a landing pad for RNA polymerase, where it just helps guide it in easier and makes transcription happen more quickly on that strand. So they can be inhibitory, they can be stimulatory, just depends. So let's talk about the bacteria gene regulation. So the whole system of this, by the way, is called an operon, which you'll see I have two definitions here. But the main idea I want you to understand when we're talking about gene regulation is that the goal is to only make exactly the products that we need, and specifically when we need them. So, for example, if I'm a bacteria and I'm doing a bunch of photosynthesis, but I'm not really eating other cells, I will be making all my photosynthesis proteins, but I'm probably not making the proteins that help me eat other cells. You basically only want to make the proteins when you need to use the protein. Otherwise, you're wasting energy, you're wasting materials. Now, it will never be perfect, but that is the goal, is to only make exactly how much we need so we do not waste energy or nutrients. Okay, so with that in mind, that we only make what we need, let's talk about this operon structure. So as a whole, an operon is a cluster of related genes that all kind of work together in some sort of feedback loop usually, and they're controlled by a single on and off switch. So they have one regulatory protein on them, and it's kind of just like a series of genes where they'll each, as you can see in this one, each catalyze a different step of this chemical reaction until you get to the final product, but they all get controlled by some other thing. So they are made up of three main things: an operator sequence, which is the segment of DNA that things bind to, same as segment of DNA that things can bind to either turn off or turn on the gene. Then there's the promoter sequence, that is the part that RNA polymerase actually attaches to. And then you have the genes, they control themselves. So the operon we're looking at on the right is the tryptophan operon, I think. It controls the synthesis of tryptophan, which is an amino acid. And so there's 5 tryptophan genes that all get activated by a single switch that tells them all to turn on so they can make some tryptophan. The fun part, well, we'll talk about this more in a second, actually, because operons come in a few main types. There's repressible operons. So this is the tryptophan example. And this is when the protein or the natural state for repressible operons is on. So by default, they will make whatever gene they code for, and they're turned off by some molecule binding to an operator that blocks the RNA polymerase from proceeding. So it doesn't prevent RNA polymerase from attaching, it just blocks it from continuing to go forward. This, in the tryptophan example, the tryptophan itself helps to regulate. So there's one of the genes that makes this regulatory protein that when it binds to tryptophan, it can attach to the RNA sequence and the DNA sequence, but it physically blocks RNA polymerase from continuing to go down along the gene. So RNA polymerase is attaching, it just can't actually go anywhere because it's blocked by this repressor. The repressor is only active when tryptophan's there. So if we go back a little bit, this self-regulates because when tryptophan is my final product of this pathway, of this operon. So when it makes enough tryptophan, it turns the pathway off by itself. If tryptophan levels are low, it won't bind to the repressor, and we won't make any tryptophan. And we will make a bunch of tryptophan. As soon as we have a bunch of tryptophan and it all binds to this repressor goes in, binds to the RNA sequence, blocks the polymerase from proceeding. Usually, repressible operons are things that consume energy. So generally like building bigger molecules from smaller ones. And it is often that the end product is actually the thing that switches it back off. The other type are inducible operons. These involve either an inducer or an activator. You'll see both names. And it's just a protein that binds to the operator sequence, which helps RNA polymerase attach. So the other one stopped RNA polymerase from proceeding. In this case, it's helping RNA polymerase attach and proceed. These ones are off by default. So if there's nothing present except the RNA strand or the DNA strand and RNA polymerase and stuff, it won't make any protein, and it needs something to activate and actually turn it on. So in this case, it's our old friend cyclic AMP, which if you remember from Unit 4 or something, was a second messenger. In this case, what cyclic AMP is doing is it's activating this cap protein, which you don't need to know what a cap protein is. It's just activating a protein that acts as an activator and allows RNA polymerase to attach to it and then attach to the DNA sequence and do its job. You can then go over the other operator sequence that is empty of a repressor. Usually inducible operons are metabolic pathways, so energy producing, taking big molecules and breaking them down, something like cellular respiration. The one we see here, and we're going to take a deeper dive into this in class, is the lac operon, which is the cluster of genes involved in breaking down lactose in bacteria. Which, if you notice in this diagram, there's an operator sequence and there is a repressor present. Sorry, something just started making a weird noise. So in the lac operon, there's actually both kinds. There's an inducible portion and a repressible portion, which is extremely common. Most of the time, an operon will have multiple regulation sites, some that promote, some that inhibit. Lets us have more control over what proteins we actually make and how much of that we actually make. So, I'm missing a slide here. I'm missing a title slide. This is the end of this video. That is another one that I wish were shorter, but I don't think there's anything we can do about it. Next video will be on eukaryotic gene regulation. I'm not expecting this to make sense on your first listen through. We will work on this in class with operons, and you should understand it much better after the assignment in class. That's all I got. Oh, wait. Oh. See ya. Bye.